Good morning, good afternoon, and good evening wherever you are in this virtual world. First of all, I'd like to give my sincere appreciation to the organizers of this conference for giving me this opportunity to briefly talk about the phenotypic and genomic architecture of indigenous African sheep. As we very well know, sheep were never domesticated in the African continent, in spite of the fact that they are very important to the inhabitants of the continent in terms of social, cultural, and economic value. However, in the continent, there is one wild caprid species that is known as the ordered, that is native to the dry Rocky Mountains in Northern Africa. The ordered has been introduced in Europe as well as in the Americas. An interesting fact is that the ordered has never been domesticated and it does not cross-hybridize with the domestic variant. Domestic sheep have a history in the African continent. In fact, the earliest evidence of sheep in Africa is derived from rock paintings that have mostly been discovered in Northern Africa, dating to about 4000 BC, both in Egypt and Algeria. These rock paintings show sheep with thin tails. In East Africa, evidence has also been found on rock paintings in Somalia and Ethiopia, and this dates to between 2000 and 1300 BC. The rock paintings found in Serekima Cave in Harar province of Ethiopia show sheep with fat tails. In Southern Africa, the evidence, however, has been very recent, and it dates to about 200 BC, and this also shows sheep with fat tails. In Africa, there are three broad types of sheep that have been described or are found in the continent. These include the thin-tailed sheep, and the thin-tailed sheep are of two varieties or of two subtypes, and this is the long-legged or Sahelian sheep and the tropical dwarf or Jalonke sheep. The long-legged or Sahelian type mostly occurs in the dry Sudano-Sahelian zone, while the tropical dwarf or Jalonke sheep occurs mostly in tropical humid regions of Western and Central Africa. Interestingly, a recent analysis of the mitogenome of these two types of sheep found two divergent groups of sheep, one representing the long-legged type and the other one representing the dwarf type, suggesting a clear maternal divergence between the two subgroups of sheep. The other group of sheep is the fat-tailed sheep, and this is the most widespread sheep in the continent, as it occurs in Northern Africa, Eastern Africa, as well as in Southern Africa. It is the only type of sheep found in Madagascar. This sheep is interesting in the sense that it depicts a very large variation in the tail morphotype, as you can see from these sheep from North Africa, these ones from East Africa, and the ones from Southern Africa. The third large group of sheep in the African continent is the fat rump sheep, and these are exclusively found in the Horn of Africa region, where they are commonly referred to as blackhead Somali sheep. This name is derived from the fact that they exhibit a very special or unique phenotype in the form of a black head and a white body. The genetic basis of these traits has not been investigated so far. Furthermore, the origin as well as the route of introduction of these sheep into the continent has not yet been investigated. In terms of phenotypic characteristics, several studies have been done on African sheep. However, there are only three that I will say are outstanding in the way they were carried out. And these are two studies, those that were commissioned by the FAO, and one study that was commissioned by the International Center for Agricultural Research in the dry area. One of the studies that was commissioned by the FAO and the one that was commissioned by ICARDA, they looked at phenotypic diversity of sheep from the Northern African region. The second study that was commissioned by the FAO looked at sheep specifically from the tropical regions of Africa. So far, these three studies are the only ones that have looked at the production systems as well as the phenotypic characteristics of African sheep in greater detail at the continental level or at the regional level. In terms of maternal and paternal genetic structure, when you compare the African sheep with sheep from Europe and Asia, we'll say that the studies of both of these paternal and paternal genetic structure are poorly investigated in African sheep. So far, there have only been 2 studies or 3 studies that have looked at mitochondrial DNA diversity, and these have mostly found the presence of haplogroup A and haplogroup B in African sheep, with haplogroup B dominating over haplogroup A. The study that was done by Meadows et al., which was looked at a global context, also analyzed some African sheep, and they found 5 haplotypes of Y chromosome in African sheep. The frequency of these 5 haplotypes were different in thin-tailed and fat-tailed sheep from Eastern and Southern Africa, suggesting most likely a contribution or differences in the paternal contribution to the ancestry of these 2 types of sheep. On the endogenous retrovirus investigations, the study done by Chesa et al. Included 1 breed of sheep from Africa, and that is the Jalonke sheep from the Gambia. The study found 1 common retroviral signature with the European primitive sheep, suggesting that there is a common history between the Jalonke and the primitive sheep from Europe. When you look at autosomal diversity, several studies have been done as well, but there is only one that is outstanding amongst all of them. And this was the study done by Anne Muigai for her PhD thesis. In this study, Anne Muigai is the only one who looked at genetic diversity and structure of sheep found across the African continent, in the sense that he used breeds from Southern Africa, Eastern Africa, Western Africa, as well as Northern Africa, and she used 16 microsatellites in her study. Muigai found out that the thin-tailed sheep from Western Africa are divergent from the fat-tailed sheep derived either from Eastern or Southern Africa. Contrary to what was expected, actually the fat-tailed sheep were subgrouped into 2 main groups, one from Eastern Africa and the other one from Southern Africa. And sandwiched between these 2 groups, there was a cluster of breeds that comprised of both fat-tailed and fat-rummed sheep, suggesting that the variation within the fat-tailed sheep found in the African continent is quite large. As I've indicated, there have been several studies done, but these have mostly been done at a country level, and the number of microsatellite markers that have been used to investigate the genetic diversity and structure have ranged between 6 and 22. I would like to point out a few of these studies, and to start us off, I would like to talk about 2 studies that looked at the diversity and structure in Algerian sheep. The first study analyzed 19 microsatellite markers in 6 breeds, and the second study analyzed 15 microsatellites in 12 breeds. The 6 breeds that were analyzed in the first study were included in the 12 breeds that were analyzed in the second study. The first study, which was done by Gaur et al., found quite some interesting results in the 6 breeds that they analyzed. They found out that 3 breeds, that is the Hamra, the Demon, and the Sidaun, show a unique or a distinct genetic profile compared to the other 3 breeds that were analyzed, which show an admixed genome profile. Now, when you look at the second study, which analyzed 15 microsatellite markers, they managed to separate, or the Algerian breeds, the 12 Algerian breeds were separated into 3 distinct genetic clusters, of which one breed, known as the Berberine, was the most distinct. The Berberine was not analyzed in the first study. However, when you contrast these 2 studies, we find some interesting findings in the sense that the Hamra, the Demon, and the Sidaun, which were found to be genetically distinct in this first study, were closely clustering together in one of the genetic clusters that was observed in the second study, suggesting close genetic relationships. Now, this finding, although it shows a contradiction, it can be attributed to the microsatellite markers that were used, in the sense that the two studies used two different sets of microsatellite markers, and therefore they could not be able to replicate the results between the two studies. This is actually not unique to this study, because this result has also been found using Egyptian sheep. These two studies, one which was done by Othman et al. And the other one done by Eltebetaji et al., also found some contrasting results, in the sense that the study done by Othman found that the Barki, the Osimi, and Rahmani had clear, distinct genetic profiles, while the same breeds, when analyzed using 13 microsatellites, they were found to have an admixed genome profile in this study. In this study by Elbetaji et al. Also showed that the Sughagi had the distinct genetic profile, while in this study, the Sughagi had an admixed genetic profile. And this clearly indicates that for most of the African studies, even when done within a country, for them to be much more informative, there is need to standardize the microsatellite markers that are used. I'd also like to talk about this third study, which was done by Gizao et al. On Ethiopian sheep, and it looked at population structure. I found this study to be a little bit unique, in the sense that apart from analyzing 17 FAO recommended microsatellite markers, they also analyzed 12 quantitative morphological traits. And the number of breeds they used were 14, and these breeds included fat-tailed sheep, thin-tailed sheep, and the fat-rumped sheep. And this is almost equivalent to what Anne Mwegai had analyzed in her PhD study. This study found out that the Gums, which is a thin-tailed sheep, was genetically divergent from the fat-tailed sheep as well as the fat-rumped sheep. But interesting enough, the fat-tailed sheep also clustered, some other fat-tailed sheep also clustered in a separate cluster. And this is also what we also observed in the Anne Mwegai study, who found two clear genetic clusters or genetic structure in the fat-tailed sheep, suggesting that there is quite a large variation in the fat-tailed sheep found in the African continent. The clustering pattern also observed with the morphological traits mirrored the pattern observed with the microsatellite markers. In recent studies, or in recent times, there have also been a few studies in African sheep that have used next generation sequencing technologies, and most of these studies have been based on genotype data that has been generated using the 50K SNP chip. One of these studies that I would like to talk about was done on Algerian sheep, and in this study, they analyzed 8 breeds using genotype data generated from the 50K SNP chip. When they did their analysis within the population, or within the country analysis, or within a country genetic analysis of the 8 breeds, they found out that the Hamra breed, the Sidaon breed, and the Tezagzawit breed were genetically distinct. But when they included all their data set in a global data set that comprised sheep populations from Italy, the Americas, and Africa, a different result was obtained. These results showed that all the 8 breeds from Algeria that were analyzed had an admixed genome background comprising of 3 genetic backgrounds. And these 3 genetic backgrounds were also observed in Italy, Americas, and African sheep. And from this study, we can say that the 2 most important lessons we learned from this is that there's a clear advantage of using SNP genotype markers, because they can be incorporated into a global database. And the second lesson is that there's importance of including, or the importance of incorporating your data set into a global database and doing a structure analysis, because it gives you a better picture of the genetic structure and composition of the breeds found within a given country. The other study I would like to talk about is on Northwest African sheep. And these Northwestern African sheep were derived from Morocco and Algeria. In this study, they analyzed 10 breeds, of which 5 came from Algeria, 2 came from Morocco, and 3 were what are called transboundary breeds. These 3 transboundary breeds are found both in Morocco and in Algeria. Now, a major finding of this study was the fact that there is a deep homogenization of the genetic profile of the 10 breeds of sheep sampled from Algeria and Morocco. The second finding was that the Sidahoun breed, the Barbarian breed, the Diman breed from Algeria, and the Tezak breed from Algeria as well showed a clear genetic profile or genetically distinct from the other populations that were analyzed. The third finding is that the Diman from Morocco, or the Diman from Algeria, and the Diman from Morocco are all characterized by different genetic background, in spite of them being one and the same breed. And this clearly suggests most likely a divergence of the breed into two different genetic groups within the same breed. The last study I would like to talk about, which looks at genetic architecture, was performed by Edwin Zomba from Southern Africa. And in this study, they analyzed 14 breeds, which included both Southern African breeds as well as commercial breeds of sheep. And they also included in their study 623 genotypes, of which 6 of these genotypes came from 6 African breeds, 2 breeds from Asia or South Asia, and 8 other breeds from Europe. And one finding of this study is that the indigenous breeds are quite different from the commercial breeds, and that they're characterized by a different genetic profile. The other thing is that the South African breeds seem to be divergent from the other African breeds, and this needs to be investigated in details. However, the level of genetic variation or the level of genetic divergence between the two seem to be not very, very large. The other finding is that all the populations can be discriminated based on either adaptation or the breed history. And these are the main discriminating factors for the breeds that were analyzed. So, as observed with the data on microsatellite markers, we find that even that this data using autosomal genetic markers or using the SNP genotype data, most of the studies are restricted in their geographic scope, and they also indicate a very close genetic relationship and high levels of genetic admixture in the breeds analyzed. Apart from analyzing genetic structure, with the development of next generation sequencing technologies has come with a very big advantage, in the sense that you can be able to assess genomic signatures of selection at the genomes of the breeds for which you are interested in. And there have been 3 studies that have been reported so far in African sheep, which have looked at genetic signatures of selection at the genome level. And 2 of these studies were done in my group, and we looked at genetic signatures for adaptation to a stressful environment. We used several selection signature analysis methodologies, and we found quite several or a large number of candidate regions that span different regions of the genome or different chromosomes. And these regions spanned a large number of candidate genes. And these, or amongst these genes which we found out, there are 3 which are worth mentioning, and this is TBS1D12, the DRHDE, and the FGF2 and the FGF9. These genes are associated with the response to environmental stress, regulation of appetite and metabolism, and melanogenesis. And these are very important traits as far as adaptation to desert-like environments is concerned. The other study I would like to mention was done or tried to investigate the genomic signatures of adaptation to high altitude environments, and this used Ethiopian sheep as the study breeds. And in this study, as we observed in our study, they also found several candidate regions, and these candidate regions spanned a number of genes, and one of the genes that is worth mentioning is the PITX1, which is responsible for cellular stress, cellular response to hypoxia. There are also other several genes that were associated or that are associated with mTOR pathway that they were found within the candidate regions that they observed. And these genes or this pathway is responsible for regulating expression of DNA damage proteins. We very well know that hypoxia and DNA damage are quite stressful factors as far as high altitude or survival in high altitude environments is concerned. Apart from those two studies, there have also been another two studies that have been published on African sheep. And one of the studies looked at genes associated with fat deposition and tail morphology in Ethiopian sheep. And this study was done in my group. And in another study, which also looked at the same, but factoring in mostly on fat deposition, was done incorporating African sheep as well as European and Asian sheep. And this study was done in collaboration between my group and the group of Elena Sciani and Fabio Pila in Italy. A common denominator in these two studies is that we found common genes which are associated either with fatty acid extrudation or fatty acid metabolism, as well as cellular fat storage. And this suggests that these could be possible candidate genes that are associated with fat deposition in the tails or in the proximity or in the tails of African fat-tailed sheep or thin-tailed sheep or fat-rumped sheep. In our study, we also found the HOXB13 gene because we used in our study long fat-tailed, short fat-tailed, and long thin-tailed sheep, and we wanted to see whether there are any differences in the tails of these sheep and what is the genetic basis of that. And we found one candidate gene, the HOXB13, which is associated with skeletal growth, and this could most likely contribute to differences in the length of the tail in the African sheep. In the study we did with Elena Sciani's group, we found this one gene, MSRB3, which was close to a QTL that is associated with tail fatty deposition, and we tend to presume this could be a possible candidate gene associated with fatty deposition. Apart from those two, there have also been other two studies that have investigated the genomics of prolificacy in African sheep. One of the study was done by Sonia Berviaf with his group or with her group in Tunisia, in which they discovered a novel mutation, which they called the fake X bar in BMP15. And they found out that this mutation is associated with increased ovulation rates in the Berberine sheep breed. In the second study, which was done on Bonga sheep, this was done in my group, and we investigated the genetic basis of prolificacy in the Bonga sheep using two selection signature analysis methodologies. And in our results, we found one selection signature on the X chromosome that spans the BMP15, and another selection signature on chromosome 5 that spans the GDF9. And we went high to suggest that these two genes could be possible candidates influencing prolificacy in the Bonga sheep. At the moment, we are in the process of either sequencing or genotyping these two candidate genes so that you can identify the possible candidate mutations. So from the few studies that you have seen that have been done on African sheep, what can we say about them, or what can we conclude about them? First of all, majority of the studies are country specific. And because of this, they tend to limit the scope of interpretation of the results that have been obtained, not only at the continental level, but also at the global level. In addition to that, we tend to presume that the increased use of dense genotyping assays, for example, the genotype data that is being generated from the 50K SNP chips or the whole genome sequence data, will most likely change this scenario because these data sets are easily amenable to integration in global databases. We also tend to presume that the formation of an African sheep omics consortia, in which case we would like to christen it as AfriSoc, could facilitate large-scale evolutionary population of functional omics studies in African indigenous sheep, and therefore shed a much more detailed picture on the genome architecture of African indigenous sheep vis-a-vis other global sheep populations. I would like to acknowledge the contribution of my colleagues to these presentations, as well as acknowledge the contribution of the CGIAR research program on livestock, GeneSeq, China contribution to ICARDA and IFAD for funding the studies that are being done in my group. Thank you very much, all of you, and please keep safe and be well. Thank you very much.